Welcome back to Principles of Organismal Biology. Today we're going to be learning about inheritance. And I have split this class into sections. One is going to be talking about inheritance at the chromosomal level, and the other one is going to be talking about inheritance at the molecular level. I would like to start this class by asking you to look at this figure and see if you can guess what's this. Okay, so hopefully you figure it out. These are chromosomes. These chromosomes are in metaphase. And you can use some props, some pieces of DNA, some information of a particular gene, maybe, that you are interested in, and you can add in this prop some fluorescent dye, so that when you hybridize this section, you can identify the position of a particular gene in a chromosome. So in this case, we have a particular gene of interest in this chromosome and another gene in this other chromosome. What allows us to get to this point? So in this class, I'm going to be introducing some concepts. What's genetics? We're going to go through some of the major experiments and studies that allow us to formulate the chromosome theory of inheritance. We're going to be differentiating between gametes and somatic cells, and we're going to determine which one is involved in inheritance, and we're going to distinguish between phenotype and genotypes. Okay, before, let's go through some definitions. And this class, as I was saying, it's going to help us to understand the chromosomal basis of inheritance. But what's a chromosome? So a chromosome is a single strand of DNA with thousands of genes. And now we know that, right? And now what's a gene? Okay, so that's the information that codes for inherited traits. And that's on a DNA molecule, okay? Now, you probably have heard the word locus, and the plural is loci. That's going to be the position of the gene in this chromosome. So here we have our chromosome in metaphase. And the loci, well, the locus is going to be a position in this chromosome. And this region is going to have a gene, a piece of DNA, a sequence of nucleotides. We're going to go through that later in the next section. And genome, this definition, the definition of a genome, it's going to be the collection of genetic information, that means the DNA in a cell. And that's all of the DNA that a cell has. Now, just for you to review this, so we have chromosomes in humans. We have 46 chromosomes. And that is 2 sets of 23 chromosomes from each parent. In this case, I am using Lynn Margulis, who is one of my heroes in biology. She was married to Carl Sagan, and they had Dorian Sagan. So they had an egg with 23 chromosomes and a sperm with 23 chromosomes. Now, these are the gametes, and they have 1 set of chromosomes. And when we have 1 set of chromosomes, we call this a haploid cell. And we notate it as 1N to say that that's 1 set. Now, somatic cells are all of the cells that we have except the gametic cells. So in all of the cells except our eggs or sperms, all of them should have 46 chromosomes. And that means 2 sets of chromosomes. And we have this in 2N or a diploid condition. So we have the two sets, remember that. We're gonna go through the Mendelian inheritance, and that it's gonna be allowing us to locate the physical basis of the behavior of chromosomes. Now, I'm gonna make a stop here, because if you are not familiar with mitosis and meiosis, I'm gonna recommend you to stop the video and look at the optional videos associated to them. Meiosis and mitosis are not covered in this class, and we assume that you know those concepts. So if you don't remember, or if you haven't been exposed to that, just watch these videos. But if you're ready, let's continue. Okay. The chromosome theory of inheritance states the following: that the Mendelian genes have a specific loci or position in the chromosomes. So that means specific genes have a position in a chromosome, and that the chromosomes undergo segregation and independent assortment. So this comes from the experiments that Mendel did. And let's go through these experiments. So Gregor Mendel was an Austrian. He was a monk, and he is known as the father of genetics. Now, what is genetics? Genetics explains how traits are passed from one individual, this is like the ancestor or a parent, to another individual or the descendant, let's say the offspring. And this is what conforms the inheritance of biological characteristics. So Mendel did some experiments. He crossed pea plants. In this case, you could see pea plants that have purple flowers and other pea plants that have white flowers. And when he crossed them, he found that all of the offspring of the F1 generation, the immediate descendants of these parents, had all plants had purple flowers. Okay, so that result suggested dominance of a trait. In this case, the dominant trait was the purple flower. The purple color was dominant, and the white flower was recessive. Okay, so then he asked, what will happen if I cross again these individuals of the F1 generation? So for his surprise, he found that the result of the crosses from F1 generation resulted in individuals with purple flowers and individuals with white flowers. So the white flower came back. How did this happen? Okay, so the other important thing is look at the ratio. So the number of purple flowers or individuals with purple flowers was a 3 per 1 of white flowers. This is key because this ratio and these results allow him to understand the basis of inheritance. And we're going to explain a little bit more on that. But he thought that there were hereditary factors, now we call them genes, that were responsible of carrying the information for each of these traits. Okay. Now, the Mendel also looked to other features. For example, we have covered the trait that he used in the flower, which was the color. But he also looked at the seeds The seeds in the peas are, you probably have eaten them. So they can be green. Most of the ones that we eat are green, but they can also be yellow, and they are smooth, but they also can be wrinkled. He also looked at the pod and some features of the pod, and he looked at the stem. And he, after all of these crosses, he concluded these three statements, and these are known as Mendel's laws of inheritance. The first one is the law of segregation. And in this case, he reasoned that the offspring will inherit one genetic allele from each parent. Okay, so one allele will go to each of the gametes, not two alleles, right? So in this case, think about yourself. You have one trait, let's say the color of your eyes, and let's just assume that's not true, but let's just assume that you have only two alleles responsible for the color of your eyes, and then one came from your mom and one came from your dad. Now think about your gametes. Your gametes are going to be the sperm or the eggs. And what this law is saying is that in your sperm or in your eggs, you're going to have only one copy, either way, the copy from your mom or the copy from your dad. Okay, let's go to the second law, the law of independent assortment. In this law, this is saying that the inheritance of one trait is not dependent of the inheritance of another trait. So he concluded that because when he did crosses, he could have independence of the ratio of the flower color, and that trait wasn't linked to, let's say, the pod, the color of the pod. So he could have white flowers with green pods or white flowers with yellow pods. So that suggests that the traits and the factors, and as I was saying, the alleles associated to these traits are independent. They are not linked. And he concluded that because he was lucky. Now we know that some traits are actually linked. So just be careful with that information. And the third law is the law of dominance. And this is the example that I was telling you, is that the purple color is dominant over the white color. Okay, so it's very simple laws that he formulated. Okay, now, if you have looked into meiosis and mitosis, then you're ready to see this slide. Okay, at the time of these experiments that Mendel carried on, we didn't know, well, he didn't know anything about alleles, DNA, genes, right? So he was talking about factors. Remember, so this is a simplified version of the meiotic process for you to put it in context. But just remember that Mendel didn't know about chromosomes. So now let's assume that we have a cross that has individuals with yellow rounded seeds, and those are big Y, and big R. So the round, it's the dominant R, and the yellow is dominant as well. And we have also individuals that are green wrinkled seeds, and they are recessive they are recessive features, both of them. So we are gonna represent them with little y, and little r. Now, these individuals are gonna be producing gametes through meiosis. And the only gametes possible that these yellow rounded seeds are gonna be forming are gametes with little big R and big Y. And similarly, the green wrinkled seeds, the individuals with these traits, are gonna be forming gametes with little y and little r, okay? So when we cross them, all of the possible combinations are gonna be individuals that have yellow rounded seeds. But now look at the genotype, right? The genotype is gonna be heterozygous, which means that we're gonna have a big Y coming from this gamete and a big R coming from the same gamete, and a little y and little r coming from this gamete from the other parent. In this case, in this generation, all of the individuals are gonna be yellow and rounded seeds. Mm-hmm. Now, this was the to explain the dominance. Now, the law of segregation in this case is going to be talking about how the alleles from each parent is going to be separated during meiosis, and at the end you're going to have gametes with only one copy. So in this case, we have a copy from the mom and a copy from the dad, let's put it that way. But in your gametes, in these gametes at least, we're gonna have independent alleles. Mm-hmm. So that's called the law of segregation. Now, what's the law of independent assortment? In this case, it is saying that big R and little r are going to be independently assorted with respect to big Y and little y, such that you could have gametes that have big R and little y, like big R and little y, or little r and big Y, or any of these possible combinations, which are 4 different combinations. If we cross these, the result, the F2 generation, is gonna provide this ratio of 9 yellow, smooth individuals per 3 of green, smooth, per 3 wrinkle, and 1 green wrinkle. The chromosome theory of inheritance proposed that the chromosomes are the carriers of the genetic material. Very simple. Now, 2 proponents of the chromosome theory exist. And one of them is Walter Sutton, an American, and another one is Theodor Boveri, a German. They work independently. Walter Sutton studied grasshoppers, and he recognized that the chromosomes came in pairs when he was looking at his at the cells of these grasshoppers. So he hypothesized that one set was maternal and the other one was coming from the dad, or was paternal. Theodor Boveri studied sea urchins, and what he noticed is that when there was an abnormal chromosome, there was an effect on the proper embryonic development of these sea urchins. Now, after this proposed of the chromosome theory of inheritance, there was more support coming from Thomas Hunt Morgan. Morgan was an embryologist, and he was working with a beautiful system, model system now, the fruit flies. And he was the one, along with his team, to provide convincing evidence that the chromosomes are the locations for these heritable factors that Mendel proposed. He used these fruit flies that had different morphological features, different traits. And this is what we call the phenotype. So the appearance, right? What is your phenotype? Well, my phenotype, I'm brown, I have long hair, I have bendy ears. You see? All of these that you could see, it's my phenotype. Now, let's go back to the flies. We have these traits in this wild type. It's the red eye, and we have a mutant phenotype. A mutant phenotype is not a wild type. The wild type is the common phenotype. And a mutant phenotype in this case, it has white eyes or albany eyes. Take-home message here is that Morgan determined that the white 8 mutant allele must be located on the X chromosome. Okay, very easy. And the second thing is that this experiment and others that Morgan did supported the chromosome theory of inheritance. Chromosomes, the number of chromosomes, the size of chromosomes are very, very different and very diverse among species. For example, some ants can have 2 large chromosomes, and we can go through all of these. Some brassicas have 18, bees have 32. Look at this. We are right here. We saw that 46 chromosomes. Chimps have 48. Now look at some of these ferns. This is a plant, a fern that has more than 1,200 chromosomes. That is a very large number of chromosomes. This is a picture of our X and Y chromosomes. But what are chromosomes? Chromosomes consist of chromatin, and that means that they are going to have DNA and proteins. And these proteins are histones. Mm-hmm. So in here we have DNA. This is the DNA in blue. And we have these groups of histones. These clusters, they have subunits, and each of these clusters has 8 subunits, and the whole complex helps the DNA to get compacted. So if you look at this, the DNA is surrounding this histone complex, and again it makes some loops, and again makes more loops, and then these loops are gonna be compacting a little bit more the DNA. So these clusters with the DNA are called nucleosomes. You can keep twisting and twisting the DNA until you reduce this very, very, very long piece of DNA into a very short space. And the maximum condensation of the DNA is going to be achieved during metaphase. This is the chromosome in metaphase. And this is the maximum condensation, right? What carries actually the genes? Right? Because we say that the chromosome is built of proteins and DNA. And on the 50s, by the 50s, we didn't know, humans didn't know if the genes were carried by the proteins or the DNA. That was a very, very important question. And that question was solved by a series of experiments that I'm going to be presenting next.